Hi there guys, and welcome to another A-level biology video. It's Mr. Phillips here, and today we're looking at synapses and synaptic transmissions. So quite a common question. It can also be linked to neuromuscular junctions, which you'll do later on when you look at muscles and then that section of the module. So then, if we look at this image here of a synapse, you can see, hopefully you're aware from GCSE almost, that there are two parts to a synapse. There is the presynaptic knob and the postsynaptic knob. So there's a swelling at the end of the neuron and then one at the start of the next neuron. And effectively there's a tiny gap called the synaptic cleft. It's not really a gap, there is obviously liquid and stuff in there that is between the two neurons. The gap is about 20 nanometers in distance. So you might think, well, what's the point of this gap? Well, we'll come to that at the end of the session. And basically action potential, so the change in charge, the electrical charge cannot jump across the gap. Instead, a chemical called a transmitter or a neurotransmitter is sent across the gap. So the impulse has to go from electrical to chemical to electrical again. And most of the time we're going to look at it when we say acetylcholine is a neurotransmitter, which is what we call a cholinergic synapse. And those are the ones that you're expected to know how they work effectively. So here we've got a little diagram which shows you a presynaptic knob, but we're going to take it forward. The key thing to note here is these calcium channels here, and we'll come back to that in a second. But before we actually look at how the information goes across the synapse, I think it's really important to look at the postsynaptic membrane first of all. So in the postsynaptic knob, you've got obviously receptor sites. So when the neurotransmitter travels across the receptor sites, there are across the postsynaptic knob, there are receptor sites that are basically built out of 5 polypeptides, and within those 5 polypeptides, the 5 proteins effectively, there are 2 receptor sites. You can see one of them in there on that diagram and one of them there. Okay. Now effectively we take a cross section of this. When there is no acetylcholine in them, they remain closed, and that stops sodium being able to go in. When acetylcholine binds to those receptor sites, and they'll have complementary shapes on the receptor sites, that opens up the sodium ion channel, and that allows then sodium to rush in, and then that can trigger depolarization, and that can reach threshold potential and then trigger a new action potential on the postsynaptic knob. So the key thing is there are 5 polypeptides, that 2 of them have got receptor sites for acetylcholine or whatever the neurotransmitter is. That causes the sodium channels to open, and then the sodium channels are then flood in. Sodium floods in, increasing the charge inside the postsynaptic membrane that causes the action potential to be fired. Now, effectively then, what this leads us to is, well, how does this whole process sort of work? So what happens is the action potential arrives. So if imagine this as an electrical impulse, which obviously we know isn't quite that, arrives at, and obviously it'll be happening across both of the membranes, either side of the axon, and it arrives at the the presynaptic knob. That then triggers calcium ion channels, which detect the change in charge, to open, and calcium ions rush in from the into the presynaptic knob from the surrounding tissue. That causes vesicles that contain the acetylcholine to fuse and complete exocytosis. So they'll move across, they'll fuse with the membrane and release the neurotransmitter into the synapse. Now the neurotransmitter then will move across diffuse across from high to low across the synapse, and they will bind to the 2 receptor sites in the postsynaptic membrane, as we already said. This causes the sodium channels to open, and then the sodium channels allow the flooding in of sodium into the postsynaptic knob, and that causes a new action potential to be fired, and that then triggers your new action potential over there. Now what has to happen afterwards, otherwise these would stay open, is that there needs to be an enzyme that breaks down that acetylcholine. So there's an enzyme called acetylcholine esterase that breaks down the acetylcholine after a couple of minutes, and then that can then go back towards the presynaptic presynaptic knob to be recycled into acetylcholine. So let's just go through it again, just to make sure we've got it in order. So the action potential arrives in the presynaptic knob, which means the sodium channels open and the ions rush in, depolarizing the membrane. That then triggers in the presynaptic membrane, the action potential triggers then calcium channels to open, and the calcium ions then rush in to the presynaptic knob. That causes the vesicles to fuse with the presynaptic membrane, and as a result that causes exocytosis, where the acetylcholine is then released into the synaptic cleft, and that diffuses across and binds to receptor sites in the postsynaptic membrane. Each action potential causes just a few vesicles to fuse, and that's enough then to trigger the new action potential at the postsynaptic membrane. But there are over 10,000 molecules of acetylcholine that are actually released. So it shows you that not a lot actually have to get across in order to fuse. And there you've got a different way of saying it here, so let's just look through this one to make sure you're completely happy. So we've got the action potential arrives, it causes the calcium ions to open up, and the calcium ions rush in, as you can see there. The calcium ions cause the vesicles to fuse in the membrane and to do exocytosis, so you can see that's happening here. That then causes the acetylcholine to be released into the cleft, and then the acetylcholine diffuses across. The acetylcholine then attaches to receptors, and the receptors then cause the sodium channels to open, and potentially potassium ions to open as well, more sodium. And then the sodium ions diffusing faster than potassium ions diffuse out, causes an action potential, and that triggers a new action potential in the postsynaptic membrane. Now, as I said, to close, the acetylcholine molecules have to leave the protein, so the acetylcholine is recycled because the synaptic cleft also contains the enzyme acetylcholine esterase, and that breaks down acetylcholine into ethanoic acid and choline, and those then get taken back across and get reabsorbed into the presynaptic membrane, where in the vesicles they reform and remake acetylcholine. You wouldn't be expected to know the details of how that happens. Now, what is the actual job of the synapse then? Well, the synapse has got lots of different jobs, because you think, what's the point of this? It slows down nerve transmission. Well, the actual main sort of jobs are, if you think about it, is it allows multiple synapses to sort of come together to one postsynaptic membrane. So you've got lots of presynaptic neurons coming together to one postsynaptic. So if you think about moving your arms, or if you detect feeling in your hands, that sends lots of impulses up your arm and to your spinal cord, and then the spinal cord then is one route into the brain effectively to allow that to happen. So other things is, if there's only one presynaptic membrane but a number of postsynaptic membranes, then you can obviously send it back out into your body as a result. Now, there are different types of synapses. There are excitatory synapses and inhibitory synapses. So neurotransmitters, like the ones we've talked about, acetylcholine is an excitatory neurotransmitter, and basically when it does bind the to the receptor, it causes an action potential in the postsynaptic membrane. However, inhibitory neurotransmitters do exist, and they can hyperpolarize the postsynaptic membrane, which prevents action potentials from being fired. So you can have some that when you get the impulse and the acetylcholine goes across, it actually inhibits the postsynaptic membrane. An example of this is acetylcholine in synapses in the heart, and when they bind to receptors in the heart, it causes potassium ion channels to open, and therefore it ends up hyperpolarizing it. And basically, a synapse where the inhibitory neurotransmitters are released are called inhibitory synapses. So in terms of other things that can affect it, drugs can be an effect on synapses, as we know. This is why alcohol makes you feel drunk, because it stops the postsynaptic membranes firing as a result, because it stops the acetylcholine from attaching to the postsynaptic membrane. But here's some examples. You can have excitatory drugs, which can have something like amphetamines, and effectively they stimulate transmitters such as noradrenaline, which increase the brain activity, and that increases the movement across the synapses, whereas inhibitory things such as beta blockers can basically block the receptors on the heart in this case, and that stops the nerves the noradrenaline can't bind.